Welcome to today's deep dive. We are so thrilled you are joining us today because we are completely rethinking a massive and honestly deeply misunderstood part of your brain.
We really are. We are talking about the cerebellum.
Yeah, the cerebellum. And to do this, we are looking at a truly groundbreaking paper. It's a 2019 study from Nature Neuroscience by King and colleagues, and it's called Functional Boundaries in the Human Cerebellum, Revealed by a Multi-Domain Task Battery.
Right. And our mission for you today is to unpack exactly how this incredibly detailed mapping project proves the cerebellum is way, way more than just a simple motor control center.
Exactly. We are going to explore how the functional boundaries of this brain region actually operate. And it completely overwrites decades of textbook anatomy.
Okay, so let's unpack this. We have to start with the historical view, I think, to understand why this paper is such a big deal.
Yeah, for the longest time, the cerebellum, which literally translates to little brain in Latin, was viewed purely as the brain's motion coordinator. That was the standard doctrine for over a century. Like if you were reaching for a coffee cup or walking down the street.
Exactly. Or playing the piano. The cerebellum was understood to be the structure making those movements smooth and coordinated. It sits at the very back and bottom of your brain, sort of tucked away under the cerebral cortex.
But over the last few decades, that simplistic paradigm has really been cracking, hasn't it?
It has. Researchers started observing clinical cases of patients who had specific damage or lesions to their cerebellum. And these patients weren't just showing clumsiness or motor deficits.
Right, they were showing distinct cognitive issues.
Yes. Advanced neuroimaging began to back this up, showing the cerebellum actively engaging in duration discrimination, attentional control, spatial cognition, emotion perception, and even complex language functions.
Which is incredibly counterintuitive when you think about traditional brain maps. I mean, we are conditioned to believe that the wrinkly outer layer of the brain, the cerebral cortex, handles all the high-level cognitive human experience stuff.
While the lower, older parts just keep us moving and breathing.
Exactly, but the researchers point out a massive problem in how we have historically studied the cerebellum. And it comes down to its physical cellular structure.
To really grasp the problem here, we need to look at what neuroscientists call cytoarchitecture. That is essentially how the cellular building blocks of the brain are arranged.
Right.
In the cerebral cortex, the cellular structure changes noticeably depending on what that part of the brain is doing. If you look at the visual cortex under a microscope, it looks physically different from the motor cortex. The cells are layered differently, the density is different. It provides physical clues about function.
But the cerebellum is frustratingly uniform. Its cytoarchitectonic organization is remarkably consistent across the entire structure.
It really is.
I like to use an analogy for this. Imagine looking at a sprawling city from an airplane where every single building is an identical gray square. You can't tell which building is the bakery, which is the bank, or which is the library because the microscopic building blocks look exactly the same everywhere.
That is a brilliant way to visualize it. Because of this cellular homogeneity, scientists needed some way to draw a map, so they relied on its macroanatomical folds.
The actual physical wrinkles.
Yeah, the cerebellum is deeply folded, almost like an accordion, along a top-to-bottom axis. Anatomists historically divided it into 10 distinct physical folds, or lobules, numbered with Roman numerals 1 through 10. For decades, researchers just used these physical folds as default boundaries.
So if brain activity happened in lobule 6, they just recorded it as a lobule 6 function.
Precisely.
Which leads to the core question of this entire study. Do these physical folds, these 10 lobules, actually represent different functional regions? Think about a wrinkled shirt. Just because there is a physical crease in the fabric, does that actually mean the shirt is serving a completely different purpose on one side of the crease versus the other?
Probably not. And to test this, the researchers had to see the brain in action.
But before we get to the test, let's do a quick recap. We want to do these little bullet point summaries every 5 minutes or so just to lock in the concepts for you.
Great idea. So bullet 1. The cerebellum is the little brain at the back of your head that we used to think just controlled movement.
Bullet 2. Clinical cases and new scans show it actually handles deep cognitive tasks like language and emotion.
Bullet 3. Because the brain cells there all look completely identical, scientists have historically just mapped it using its physical folds, called lobules 1 through 10.
And our goal today is to figure out if those physical folds actually mean anything for brain function.
Perfect. So to figure that out, the researchers created what is arguably the ultimate brain gauntlet. They developed a multi-domain task battery, or MDTB.
the MDTB, and the sheer scale and rigor of this experiment are what make the findings so undeniable. They took 24 healthy participants and put them through a battery of 26 completely diverse tasks.
And then these were not simple tasks. They were broken down into 47 unique conditions covering a massive spectrum of human experience. They wanted to test motor control, cognitive processes, affective or emotional processing, and social processes.
They really threw everything at them. They had these participants doing complex math, recalling autobiographical memories from their childhood, watching movie clips, tapping their fingers in very specific sequences.
They viewed highly emotional images and read complex narratives. They wanted to fire up every conceivable circuit in that little brain.
The preparation for this is where the methodology really shines for me. Before anyone was allowed near the brain scanner, they had to go to training camp.
Yes, the 3-day behavioral training.
They spent about 10 hours total practicing all these tasks. I found this detail totally fascinating. Imagine having to practice tapping your fingers and doing mental math for 3 days just to prepare for a brain scan. Why put them through all that grueling prep work?
It comes down to isolating the exact variable you want to measure. When you learn a new task, your brain activity is chaotic. It is dynamic. It requires immense effort, and different networks are constantly firing as you figure out the rules.
You're mapping the struggle, basically.
Exactly. The researchers did not want to map the process of learning. They wanted to map the actual steady-state function of the cerebellum during seamless execution.
That makes so much sense.
Right. So by day 3 of training, the participants were hitting at least an 85% accuracy rate across all the tasks. The tasks had become automated. Only then did the actual scanning begin.
And the scanning was a marathon. They used functional magnetic resonance imaging, or fMRI. Just as a quick refresher for you, an fMRI scanner is a massive magnetic tube that tracks blood flow in the brain.
Because when a specific brain area works harder, it needs more oxygen.
Right, so blood rushes to that area. The fMRI tracks that blood flow to create a real-time map of brain activity. Each person spent a staggering 5.5 hours inside this scanner, broken up across 4 different sessions.
And to make sense of the mountain of data this generated, they used a very strict block design. The block design is essential for making clean comparisons.
How does a block actually work in this context?
Each task was performed in a specific 35-second block. The first 5 seconds provided the instruction, so the screen might say, "Theory of mind task, read the following story."
Okay.
That was immediately followed by 30 seconds of continuous task execution. Because every single task was measured in these identical blocks against a shared common baseline, the researchers could make direct apples-to-apples comparisons.
So if you want to know exactly how your brain's blood flow response to watching a sad movie differs from its response to doing mental math, this setup gives you that precise mathematical contrast.
Exactly.
Let's do another quick recap of this setup for you. We have 24 highly trained people, 26 diverse tasks, and over 5 hours of high-resolution blood flow tracking per person. Now we arrive at a really critical math problem. How do we actually know if a boundary in the brain is real?
This was the major hurdle. They had to invent a new mathematical algorithm, basically, to prove it. They called it the distance-controlled boundary coefficient, or DCBC.
The DCBC. It sounds incredibly technical, but there is a great analogy we can use to visualize it. Imagine the brain is a sprawling city.
Okay.
The fMRI scanner gives us data in voxels, which are essentially 3-dimensional pixels of brain tissue. Let's imagine those voxels are individual houses in this city. If a functional boundary in the brain is real, like a physical wall separating 2 totally different neighborhoods, then 2 houses inside the exact same neighborhood should behave much more similarly than 2 houses that are separated by that boundary wall.
Even if the physical distance between them is exactly the same.
Exactly.
And the DCBC algorithm calculates exactly that spatial relationship. It measures the correlation of the activation patterns between pairs of voxels. If a boundary is functionally meaningful, the correlation between 2 voxels within a defined region should be significantly higher than the correlation between 2 voxels spanning the boundary.
And the brilliant part is that the formula strictly controls for the spatial distance between the voxels. So you aren't just measuring the fact that things closer together act more similarly.
It is a brilliant metric. And the researchers took this shiny new DCBC math and applied it to the traditional textbook map.
Right, those physical folds, lobules 1 through 10, that anatomy students have been memorizing for decades.
And here's the big reveal. When they tested those physical boundaries, the correlation difference was tiny. The DCBC score was just 0.03.
Wow, 0.03. So to put it bluntly, it means the physical fissures in the cerebellum do not demarcate a change in function.
They don't. The traditional map is functionally weak. The paper highlights a specific glaring example. There's a physical boundary separating two prominent areas known as Crus and Crus.
Crus and Crus, yeah.
When they ran the DCBC on that specific boundary, it did not predict any functional specialization whatsoever. The score for that boundary was literally 0.
A flat 0. The wrinkly folds are just wrinkles. They tell us almost nothing about what the underlying tissue is actually processing, so the researchers literally tossed out the old map.
They had to. Since the physical landmarks were useless, they used their massive MDTB blood flow data set to draw a brand new data-driven map. They let the function dictate the borders, and they ended up dividing the cerebellum into 10 distinct functional regions.
10 regions instead of 10 physical lobules. And the statistical performance of this new 10-region map was striking. When they tested it using the DCBC metric, it achieved a score of 0.130.
Which vastly outperforms the anatomical map. But the true test of any scientific model is cross-validation. A map is only genuinely useful if it can predict boundaries for a completely new set of tasks that it was never trained on.
And it did exactly that. It successfully predicted the functional boundaries for entirely new cognitive and motor tasks.
It is incredibly robust.
Time for another quick recap before we dive into what I think is the coolest part of the study. Bullet 1. The researchers ran a massive 35-second block design gauntlet of tasks.
Bullet 2. They invented the DCBC algorithm to prove if a boundary is real, comparing voxel houses in the same neighborhood versus across a wall.
Bullet 3. The old physical folds scored a near 0, proving they are basically a lie when it comes to function.
And bullet 4. They drew a brand new, highly accurate 10-region functional map.
So let's take a tour. What do these 10 regions actually do?
Let's start with the motor features. Regions 1 and 2 are relatively straightforward. They were driven almost entirely by left-hand and right-hand movements, respectively.
They handle the classic sensory motor coordination we've always associated with the cerebellum.
Right. But Region 3 yielded a massive, paradigm-shifting surprise. Region 3 is centered around an anatomical area called vermis 6.
Vermis 6, and prior to this study, vermis 6 was the subject of intense debate, wasn't it?
It was. A lot of older neuroimaging studies suggested this area was related to emotional processing because it frequently lit up when participants were doing emotional tasks.
But this study completely debunked that by doing something very clever. They strictly tracked eye movements.
This is why you utilize a multi-domain task battery. It exposes the flaws in narrower studies. Previous researchers might have seen vermis 6 light up while a participant was looking at a sad picture, and they concluded, vermis 6 processes sadness.
But by tracking eye movements across all 26 tasks, this study proved definitively that Region 3 is an oculomotor region.
Yes, it is driven by saccades, which are the rapid darting movements your eyes make as they scan an environment.
They proved it wasn't an emotion center because when they gave participants passive emotional tasks like just staring fixedly at sad faces without scanning the image, Region 3 barely activated at all.
It only lit up when the eyes were darting around under high attentional demands, like searching for Waldo in a complex picture.
If older studies didn't track eye movement, they were basically flying blind and misinterpreting the data.
It is a perfect example of why controlling for multiple variables simultaneously is crucial in neuroscience. So moving outward from those primary motor and eye movement areas, we find Region 4.
Which is a massive area surrounding the hand motor regions. And what is fascinating here is that this region was highly activated not by moving, but just by watching action.
Yes, action observation. It suggests that Region 4 acts as a premotor, or simulation area.
This is such a relatable phenomenon. Have you ever watched a really tense sports game, and when a player goes to kick the ball, your own leg twitches?
Or you see someone stub their toe, you physically wince.
Exactly. That is Region 4 of your cerebellum actively firing. You are simulating the movement in your own mind just by observing it, helping you comprehend the action and maybe even empathize with it.
It's heavily engaged in action planning and comprehension. But the real revelation of this 10-region map lies in the purely cognitive areas. Regions 5 through 10 make up a huge portion of the human cerebellum, and they have absolutely nothing to do with physical movement.
Let's break these down because this is where the little brain really shows off its processing power. Regions 5 and 6 are highly associated with divided attention and working memory.
So when you are walking down a busy, noisy street, while simultaneously trying to keep a 7-digit phone number in your head, regions 5 and 6 are doing the heavy lifting to keep those items active in your mind without dropping them.
Then we move to regions 7 and 8, which are specialized for narrative and word comprehension.
As you, the listener, are hearing our voices right now, processing the structure of these sentences, and understanding the overarching story we are telling about the brain, regions 7 and 8 in your cerebellum are highly active. They are working in tandem with the language centers in your cerebral cortex.
And region 9 takes it a step further. It is deeply involved in active language processing, like verbal fluency.
When you are playing a game of Scrabble, or you have that tip of the tongue feeling where you are desperately searching for the exact right word to use in a conversation, region 9 is actively engaged in that linguistic search.
Finally, we reach region 10, which sits at the very outer lateral edges of the cerebellum. This region is dominated by autobiographical recall.
It is actually a crucial node in what neuroscientists call the brain's default mode network.
The default mode network is so fascinating. It's the network that hums to life when you stop focusing on the outside world. When you are staring out a bus window, daydreaming, remembering what your childhood kitchen smelled like, or imagining what you were going to eat for dinner tomorrow, that is your default mode network.
And region 10 of the cerebellum is right there orchestrating those internal mental time travels. It fundamentally changes our appreciation of this structure.
It totally does. But it also brings up an important methodological debate in brain mapping. Before this MDTB study, many scientists attempted to map the cerebellum using resting state fMRI.
Right. Instead of having people perform tasks, they just put them in the scanner, told them to relax, let their minds wander, and track the spontaneous baseline fluctuations in blood flow. They wanted to see which areas naturally pulse together.
And this study actually tested their new map against those resting state maps. They compared the task-based boundaries to the task-free boundaries.
And the result was decisive. The MDTB task-based map significantly outperformed the resting state maps in predicting true functional boundaries, particularly in the lateral areas engaged in motor control and action observation.
The underlying mechanism for why this happens makes logical sense, doesn't it?
It does. Spontaneous resting state fluctuations are incredibly useful for identifying broad, general networks, like identifying the entire default mode network across the whole brain.
But they struggle to identify sharp, specific functional boundaries between adjacent regions.
Exactly. To truly map the precise borders of function, you have to see the brain engaged in specific demanding actions. You have to force the systems to differentiate themselves under load.
It's time for our final mini recap. We toured the 10 regions. We have hand movements, eye movements, action observation, working memory, narrative comprehension, language processing, and daydreaming, all packed into the cerebellum.
And we learned that you have to give the brain an active task to actually map these borders clearly. Just resting isn't enough.
Which brings us to a statistic in this paper that absolutely demands our attention. It has to do with how unique you are.
The researchers analyzed how similar these functional maps were across different participants. Given that we all share the same basic human anatomy, you would logically assume our functional brain maps would be near identical.
But the study found something astonishing. While 27.7% of the brain pattern variance was shared across individuals, a massive 72.3% of the fine-grained functional patterns were entirely idiosyncratic.
Meaning they were completely structurally unique to the individual participant.
72% of your functional map is uniquely yours. To put that in perspective, the 27% that we share is like the general zoning of a city. We all generally have the language neighborhood and the motor neighborhood in the same rough geographic area of the cerebellum.
But the 72% that is unique to you represents the exact property lines, the specific shape of the streets, the exact borders of those functional neighborhoods. Your high-resolution cerebellar map is as unique as your fingerprint.
The practical implication of this is huge for neuroimaging. The researchers demonstrated that when they derived a parcellation map, a boundary map, using only data from one specific individual, that highly personalized map vastly outperformed a generalized group average map when predicting functional boundaries for that specific person.
A one-size-fits-all brain map is inherently flawed when you look closely enough.
So we have this highly individualized, invisible functional map dictated by blood flow. But how does that invisible map connect to the actual physical meat of the brain? Earlier, we talked about cytoarchitecture, the fact that the cells look uniform everywhere. How do these functional boundaries relate to the microscopic biology?
This requires us to look at the wiring of the cerebellum. While the basic cellular structure looks uniform, advanced molecular techniques have revealed hidden layers of organization. For example, the cerebellum has these things called Zebrin to second zones.
Zebrin the second zones. Let's create a visual memory trick for that. Imagine painting invisible chemical stripes, almost like zebra stripes, directly onto the brain cells of the cerebellum.
That is essentially what they are. These chemical stripes dictate how certain incoming neural wires connect. Specifically, a type of input called olivocerebellar projections perfectly respect these rigid striped boundaries. They wire themselves strictly according to the zebra stripes.
But here is the catch, right? The functional boundaries we see with the fMRI blood flow tracking do not align perfectly with those neat zebra stripes.
Correct. And that is because the fMRI signal in the cerebellum is primarily driven by an entirely different set of incoming wires called mossy fibers. Unlike the rigid inputs that follow the stripes, mossy fiber inputs are messy. They are diffuse and patchy.
Imagine grabbing a handful of cooked spaghetti and throwing it against the wall. It sticks in irregular, patchy clusters. That is your mossy fiber wiring system.
It's an apt, if messy, analogy. These mossy fibers likely span multiple molecular zebra stripes. So the highly unique, idiosyncratic functional map we are seeing, that 72% of uniqueness, is likely a direct reflection of how your specific, patchy, mossy fiber connections wire themselves as you developed and learned.
We are coming to the end of our deep dive, so let's synthesize the journey we've taken today. We started with a cerebellum that was historically viewed just as a motor controller, mapped by physical folds that turned out to be functionally meaningless.
We explored a massive multi-domain task battery that put 24 people through an absolute gauntlet of cognitive, motor, and social tasks.
Through rigorous block design fMRI and the innovative DCBC algorithm, we saw the old physical map discarded. In its place, the researchers drew a 10-region functional map that proves the cerebellum is a massive cognitive multitasker, handling everything from eye tracking and working memory to language processing and autobiographical time travel.
We also learned that to find these borders, the brain needs to be active. Resting state scans just blur the lines. And perhaps most astonishingly, we discovered that your functional map is overwhelmingly unique to you, driven by the patchy, idiosyncratic wiring of your mossy fibers.
And we must emphasize that this new map is not just academic trivia. It is a vital, necessary tool for the future of medicine.
It really is. There is growing evidence that cerebellar dysfunction is involved in a wide range of neurological and psychiatric disorders, including schizophrenia, autism spectrum disorder, and dyslexia.
Having a functionally defined, highly precise map allows researchers to pinpoint dysfunction in specific cognitive regions of the cerebellum rather than just guessing based on physical folds. It helps us understand the complex circuits that dictate how we process the world.
It gives medicine a much-needed, highly accurate compass.
It does. But before we sign off, I want to leave you, the listener, with a final, provocative thought to mull over. We learned today that a staggering 72% of the fine-grained functional map of your cerebellum is uniquely yours. It is your own personal neural fingerprint, shaped by the patchy spaghetti of your mossy fibers.
A truly individual map.
So consider this. If the physical way your brain processes language, holds attention, and recalls memories is structurally unique to you at a functional level, how much of what we consider your distinct personality or specific sense of humor or your unique talents is just the idiosyncratic way those mossy fibers decided to wire themselves across these 10 regions?
Are the things that make you you just the result of a highly personalized invisible map operating quietly at the back of your head?
It is absolutely something to think about. Thank you so much for taking this deep dive with us.